Welcome to the MedPod, everyone. Get ready, because today we are diving deep into the world of molecular cell biology.
Ooh, yes.
We had tons of requests from you guys for a solid overview of the central dogma.
The central dogma.
So that means we're going to be hitting DNA, RNA, protein synthesis, and a little peek at some of the tools that scientists use to study all of this.
It's going to be, it's really going to be like unlocking the secrets of life.
Right.
You know, how does information actually flow from the DNA to RNA to protein? How do we regulate that? And what happens when things go wrong?
Right.
Which, let's face it, is sometimes how we get diseases.
Yeah, so let's start with the absolute basics here.
Okay.
What are we even working with? Like, what are the fundamental building blocks in this molecular world?
Well, you know, if you want to think about it like building a house,
Okay.
you can't do it without bricks and mortar.
Right.
And the same goes for a cell, right? We need those basic components, nucleosides and nucleotides.
Oh, yeah.
And these are what actually make up the DNA and RNA, those genetic blueprints.
So I'm hearing kind of like the letters of our genetic alphabet.
Yeah.
But what makes these molecules so special? What can they do?
Well, I think it's really interesting that they're not just these passive little blocks, right?
Right.
They actually play really important roles in things like storing energy in the cell, signaling,
Oh,
and even how enzymes function.
okay.
For example, ATP, and you've probably heard of that, the cell's energy currency that's a nucleotide.
Wow, okay.
Yeah.
So we got the building blocks, but how do they all come together to actually make these structures of DNA and RNA?
Okay, so let's start with DNA.
Correct.
It's not just like a random pile of nucleotides.
Right.
It's got that beautiful structure, like a twisted ladder, the double helix and the
Yeah.
rungs of that ladder.
Yeah.
Those are formed by those complementary base pairs, adenine with thymine,
Okay.
and cytosine with guanine.
Right, the famous ATCG.
Exactly.
So it's kind of like they're dancing, right?
Yeah, I like that.
With a specific partner.
Yep. Exactly. They've got their match. And this pairing, it's super important for DNA replication,
Okay.
because that makes sure that when we're passing on genetic information, we're doing it accurately.
Gotcha.
But there's another important detail.
Right.
The two strands of DNA actually run in opposite directions.
Oh,
It's called anti-parallel orientation.
okay.
So if you imagine like a two-lane road
Right.
with traffic going in opposite directions, but somehow it all works together.
Gotcha. Okay, so we have our DNA blueprint,
Right.
but how do we actually use this information to build all the machinery of the cell?
That's where RNA comes in.
Oh,
Because RNA is kind of like the messenger molecule, right? It carries the genetic information from DNA to the ribosomes.
Okay.
And the ribosomes, those are the protein synthesis factories of the cell.
right. So that's where it all comes together.
Exactly.
And there are like different types of RNA, right, involved in all of this.
You're absolutely right.
Yeah.
So we've got messenger RNA, mRNA, which carries the code from DNA to the ribosomes. Ribosomal RNA, or RNA, which is actually a component of the ribosomes themselves.
Oh, wow.
And then we've got transfer RNA, tRNA, which acts like a delivery truck.
Yeah.
It brings the correct amino acids, and those are the building blocks of proteins, to the ribosomes based on what the mRNA says.
Wow, that's very organized.
It is. It's like a well-choreographed ballet.
Yeah.
Every molecule is doing its part.
So it's not just those three.
You're catching on.
Okay.
No, there's actually a whole world of what we call non-coding RNAs.
Okay.
Or ncRNAs. And these guys, they don't directly make proteins,
Ugh.
but they're really important in regulating gene expression.
So like, what do they do?
Well, imagine they're like the stage managers
Okay.
of this whole production, right? They're controlling the lighting, the sound, who gets cast.
Wow, so even though they're not making proteins, they're still calling the shots.
Exactly. They're the fine tuners.
So they're still essential.
Absolutely. And we're constantly discovering new and exciting roles for these ncRNAs. Some are involved in silencing genes, some are fighting off viruses,
Oh,
and
wow.
we're even starting to see some that could have therapeutic potential.
It's like a whole hidden world.
It really is, yeah.
Okay, so let's move on to the stars of the show then. The proteins.
All right.
These are kind of the workhorses, right?
They are. They do it all.
That get everything done.
Yeah, and there's so much diversity in proteins.
Yeah.
And their structure is so closely related to their function, you know?
Okay.
They can fold up into these really complicated 3D shapes,
Right.
from simple spirals to these crazy tangles, and that's what allows them to do such specific jobs.
So each one kind of has its own like personality and skill set.
Yeah, absolutely.
Okay, so how do we actually get from the information in DNA to a protein?
That's the central dogma of molecular biology, my friend.
Okay.
The flow of information from DNA to RNA to protein,
Right.
it's the foundation of life.
So let's break down the central dogma then.
Yeah.
So the first step is DNA replication. Right.
We got to copy that DNA molecule first.
Right.
So each new cell gets a full set of those instructions.
It's like making a copy of the blueprints.
Exactly. Yeah.
And I remember that DNA replication is super accurate
It is.
because of those proofreading enzymes.
Yes. DNA polymerase, that's the enzyme that does the copying.
Right.
It has these built-in proofreading mechanisms.
Wow.
So it can catch those errors.
It minimizes the error.
Exactly. But even with that, sometimes mistakes happen.
Okay, so what happens then?
Why?
Does the cell just like scrap the whole blueprint?
Not necessarily.
Okay.
Cells are pretty clever.
Okay.
They've got sophisticated DNA repair mechanisms to fix those mistakes. So it's like having a team of editors going over the blueprint and making sure everything's perfect.
Okay, so multiple levels of quality control.
Absolutely, because those repair mechanisms are crucial for stopping those mutations that can lead to diseases like cancer.
Yeah, no, that's good.
Yeah.
So the DNA has been replicated. What happens next in the central dogma?
Next up is transcription, which is where we convert the DNA into RNA.
Okay.
It's like taking that handwritten document and typing it up.
Right, so the DNA unzips again.
Yeah.
And this time, we have an enzyme called RNA polymerase.
Yes.
And it's reading one of those DNA strands.
Yes.
And creating that complementary RNA molecule.
It's like RNA polymerase is taking dictation from the DNA
Yeah.
and making that RNA transcript that carries the code.
Okay, so that transcript carries the instructions to make proteins.
Exactly. But in eukaryotes, and that includes us, there's an extra step.
Oh, okay.
It's called RNA processing.
Okay.
It's like editing that rough draft of the transcript before it goes to print.
So we're adding the cap at the beginning, the tail at the end.
Yeah.
And we're taking out those non-coding bits, those introns.
Right. We got to leave the exons, those protein coding parts.
Right. Right. And the splicing.
Oh, the splicing.
Can get kind of funky, right?
It can. It's not always straightforward.
Okay.
We can include or exclude some exons, and that's called alternative splicing.
Okay, wait. So a single gene can actually make different proteins.
It can. It's wild.
That's really cool.
It's like the molecular version of a choose your own adventure book.
Yeah.
So we can get this huge diversity of proteins, even with a limited number of genes.
Oh, okay. So we have our edited RNA transcript.
Yes.
Ready to be translated into a protein.
Ready to go.
What happens next?
That, my friend, is the final step of the central dogma translation. And this is where we convert that RNA code into a protein.
So this is all happening at the ribosomes.
Exactly. Back to our protein synthesis factories.
Okay, so the ribosomes are reading the code.
Yep.
3 nucleotides at a time, the codons.
You got it. And each codon says which amino acid we need. And remember, amino acids are the building blocks of proteins.
Right, right.
So the tRNA molecules, our delivery trucks, they bring the right amino acid to the ribosome based on what the codon says.
Okay, and then the ribosome links them all together.
Yep, forms that polypeptide chain, which will fold up into a functional protein.
So it's like a little molecular assembly line.
I like that.
Each tRNA bringing the next part.
Right, all happening based on those instructions from the DNA.
Wow, that's really cool.
It's amazing.
It's pretty remarkable that all of this is happening
all the time.
in every cell of our body, constantly.
Constantly.
Okay, so how do scientists actually study this? What are the tools that they use to kind of like unravel all this?
That's a great question because scientists, they have this whole arsenal of tools that they use to study DNA, RNA, protein.
Right.
And these tools have totally revolutionized how we understand molecular biology.
Okay.
And they've opened up so many doors for research and developing therapies.
So that sounds like the perfect place to pick up next time.
I think so, yeah.
I'm really interested to learn about these molecular detective tools and how they're used to kind of like decode the cell.
It'll be fun.
So join us next time for part 2 of our deep dive into the world of molecular cell biology.
Welcome back to the MedPod. So before the break, we were just about to jump into the world of molecular tools.
Yeah, I'm really excited about this.
Yeah, it's cool stuff.
It's like we're getting a detective's toolkit for the microscopic world.
Exactly. Yeah.
So what can you tell me about these tools and how they've changed the game for molecular cell biology research?
Okay, so one of the things that really shook things up early on was the discovery of these things called restriction endonucleases, which are basically enzymes that act like molecular scissors.
What?
Yeah, they cut DNA at specific sequences.
Oh, wow. So scientists can use these to cut and paste DNA fragments. Like become genetic engineers.
Exactly. That's what it opened up.
Wow.
This whole world of genetic engineering.
That's incredible.
Yeah, manipulating DNA in ways that, people couldn't even imagine before.
Right.
So being able to create those recombinant DNA molecules, that's the basis for so much biotechnology.
That's so cool.
It is.
But working with these tiny DNA fragments, I imagine that's got to be a challenge.
So how do you amplify that, right?
Yeah.
So you can study it.
Yeah.
Well, that's where PCR comes in.
Oh, yeah. PCR.
Polymerase chain reaction.
I've heard of that.
It's like a molecular photocopier.
Okay.
You can take a teeny tiny DNA sample
Right.
and make tons of copies of a specific sequence.
And that's used for so much.
It is. It's everywhere.
Like disease diagnosis, forensics.
You name it.
Yeah.
It's crazy that something so simple can be so powerful.
Right. Okay, so once scientists have amplified these fragments, how do they actually read the genetic code?
Well, that's where DNA sequencing comes in.
Okay.
And sequencing, oh my gosh, technology has just exploded from that classic Sanger method to these super high throughput platforms like Illumina sequencing.
So now we can sequence entire genomes really quickly and it's not that expensive.
Yeah, which has opened up like a whole new world for genomics research.
It's crazy how far it's come.
It really is, yeah.
Okay, so I remember you mentioned something called cDNA cloning before.
Oh, yeah.
What is that exactly?
Okay, so cDNA cloning, it's a way to make DNA copies of mRNA molecules.
Right, and mRNA is basically the genes that are being expressed, right?
Exactly, so it's like a snapshot of what's happening in the cell at that moment.
Oh.
So by creating these cDNA libraries, scientists can really focus on the genes that are turned on in specific cells or under certain conditions.
So it's a way to kind of narrow things down.
Exactly.
Okay, that makes sense.
Yeah, super useful for studying gene expression patterns.
Okay, so shifting gears a bit.
Yeah.
What about manipulating gene expression? Can scientists turn genes on or off?
Oh, absolutely.
Really?
Yeah, and we call that gene expression inhibition.
Okay.
And it's a super powerful tool for figuring out what genes do, and even for developing therapies.
So how does that work?
Well, one example that I think is really cool is RNA interference, or RNAi.
Okay.
It's like a guided missile system for silencing genes.
Okay.
So you introduce these little molecules called siRNAs into the cell, and they target and destroy specific mRNAs.
So you're basically turning off that gene.
Exactly. It's like a molecular off switch.
Wow, that's incredible.
It is, and it's got huge potential for both research and therapy.
Okay.
People are using it to study gene function, find new drug targets, and even develop treatments for things like cancer and viral infections.
That's amazing. It seems like there's a tool for everything.
There really is.
But how do scientists actually study the proteins themselves?
Oh, good question, because they're the workhorses, right?
Yeah.
There are tons of techniques, but two really common ones are immunoprecipitation and Western blotting.
Okay, I've heard of Western blotting. Isn't that where you use antibodies to detect specific proteins?
Bingo. You separate the proteins by size on this gel.
Okay.
And then you can visualize them.
Okay.
And that tells you how much of a specific protein is there, and even if it's been modified.
Okay, and what about immunoprecipitation?
So immunoprecipitation, it's like fishing. You're using antibodies to pull out a specific protein from a mixture.
Okay.
So you can really zero in on it.
So with these tools, scientists can study protein interactions, signaling pathways, and even post-translational modifications.
Exactly.
Wow, we've come a long way in our ability to study this molecular world.
We have.
It's pretty remarkable.
It is, and it's given us such incredible insight into how cells work.
Yeah?
And opened up all these possibilities for research and therapy.
And new tools and techniques are being developed all the time.
Oh, yeah, it's a really exciting time.
So I feel like we've been zoomed in on the molecular level for a while now.
Yeah.
Maybe it's time to zoom out a bit and look at the cell itself.
Yeah, I like that.
That bustling metropolis of organelles and pathways.
All right, let's take a tour.
I'm ready.
Let's go into the cell and explore some of its key organelles, those specialized compartments that make everything happen.
Perfect. I'm ready to explore the cellular city. Welcome back to the MedPod. It feels like we've been on this incredible journey from the blueprints of life to the construction sites where proteins are made.
We have, and now we're about to enter the city itself, the bustling city of the cell.
Yeah, I love that analogy of the cell as a microscopic metropolis.
It works so well.
You've got all these different districts specializing in different things like energy production, waste management, transportation.
Exactly. And just like any city, a cell needs a strong infrastructure.
Right. Okay, so where should we start our tour?
Well, how about the nucleus?
Oh, the nucleus, yeah. The command center.
Exactly. That's where the DNA blueprint resides.
So it's kind of like a vault, right?
Yeah, protecting the city's most valuable treasures.
But also controlling access to those blueprints.
Absolutely. And it's not just storage.
Okay.
The nucleus is also where DNA replication and transcription happen.
Right. Those processes we talked about earlier.
Exactly. Essential for passing on genetic information and making those RNA transcripts.
So the nucleus is like a hub of activity.
Constantly churning out instructions for building and maintaining the cell.
Okay, so what's next on our tour?
Well, how about the construction zone?
Okay, the construction zone.
The endoplasmic reticulum, or ER.
Ah, the ER, right, where proteins and lipids are made.
Exactly.
I always picture it as this network of folded membranes, kind of maze-like.
It is, and it's where those ribosomes, our protein-making machines, do their work.
Right, the ribosomes. So is that what makes the rough ER rough?
Exactly. The rough ER is studded with ribosomes.
Oh, okay.
It's where protein synthesis happens, especially for proteins that are going to be secreted from the cell or incorporated into membranes.
So like the rough ER is the factory churning out these essential proteins.
Exactly. But the ER has another side, the smooth ER.
Smooth ER. What's happening over there?
Well, it's smooth because it doesn't have ribosomes.
Okay, so if the rough ER is the factory, what's the smooth ER's specialty?
Ooh, good question. The smooth ER is important for lipid synthesis, detoxifying harmful substances, and storing calcium.
Wow, so it's like a multi-purpose facility.
Exactly, like a refinery, a waste treatment plant, and a storage facility all rolled into one.
Okay, that's amazing. So we've got proteins being made in the ER.
Right.
Where do they go next?
To the Golgi apparatus. Think of it like the city's postal service.
The Golgi, so it sorts and packages proteins for delivery.
Exactly. It modifies them too.
Oh, okay.
The Golgi is a stack of these flattened membrane sacs, and it's like a series of processing and sorting stations.
Wow, very efficient.
It is. The proteins move through the Golgi, get modified, and then they're packaged into these little vesicles for transport.
So it's like a logistics network.
Yeah, making sure every protein gets to its destination.
Very cool. Okay, so we can't forget about waste management, right?
Oh, absolutely. Every city needs that, and so does the cell.
Yeah, so how does the cell deal with its waste?
Lysosomes.
The lysosomes. Those are the recycling centers.
Exactly. They're like these little bags of digestive enzymes.
Right, that break down all the waste and debris.
And even worn-out organelles. They recycle those valuable components.
So it's like the city's sanitation department, keeping everything clean.
Absolutely. Now, to keep this whole city running, we need energy, right?
Right, lots of energy.
That's where the mitochondria come in.
Oh, the mitochondria. The powerhouses.
For powerhouses?
Yeah, those bean-shaped organelles with all those folds.
Yeah, those folds are called cristae.
Okay.
And they increase the surface area for cellular respiration.
Right, that's how we convert nutrients into ATP.
Exactly, the cell's energy currency.
So mitochondria are like the power plants of the cell.
Keeping everything running. And you know what's really interesting?
What?
Mitochondria have their own DNA. When ribosomes.
Wow.
It suggests they were once independent organisms.
No way.
Yeah, that formed this symbiotic relationship with early cells.
Wow, that's incredible. Like a little city within a city.
I know, it's fascinating. Okay, so we've got all these organelles, but they need to be organized and supported.
Right, of course, like the scaffolding of the city.
Exactly. That's where the cytoskeleton comes in.
Okay.
It provides structural support, helps things move around.
So it's dynamic, it can change?
Oh, yeah. It's constantly adapting to the cell's needs.
Okay.
And it's made up of these protein filaments, microfilaments, microtubules, intermediate filaments.
So it's like a network of roads and bridges.
Exactly.
Yeah.
Facilitating movement and communication within the cell.
Very cool. So we've talked about all these internal structures.
Right.
But cells don't exist in isolation, right?
Of course not. They
Right.
need to interact with their surroundings.
Yeah, receive signals, exchange materials.
That's where the cell membrane comes in.
Right, the cell membrane, like the city walls.
Yeah, but it's more than just a wall.
Oh.
It's selectively permeable. It controls what goes in and out.
Oh, right. It's made up of lipids, proteins, carbohydrates.
Exactly. And it's how the cell talks to the outside world.
Okay, that makes sense.
Yeah. And don't forget about cell junctions.
Oh, right. Cell junctions, like the gates and bridges of the city.
Exactly. They connect cells together so they can communicate,
All right.
exchange materials, and form tissues and organs.
So there are different types of junctions.
Yeah, like tight junctions, they create a seal between cells so nothing leaks out.
Okay.
And gap junctions, they form channels so cells can talk directly to each other.
It's amazing how cells have evolved these complex ways to communicate and cooperate.
It is. It's like a perfectly orchestrated symphony.
It really is.
And all of this is happening in every cell of our bodies.
All the time.
All the time.
Mind-blowing.
It really is. And we've only just scratched the surface.
I know, this deep dive into molecular cell biology has been incredible.
I'm glad you enjoyed it.
I feel like I have a whole new understanding of how cells work.
It's a fascinating world.
It really is. Thank you so much for joining us on this episode of the MedPod.
It was my pleasure.
And to all our listeners, we'll catch you next time for another deep dive into the world of science and medicine.